Thanks very much to the organising committee for inviting me to speak. It's a real pleasure to be here. Thanks to Dave as well for obviously inviting me. And yeah, great conference as well. So I'm going to talk to you about solid state biological aerosols. I'm really going to focus on the in silico tools and methodologies that we've been developing to try and shortcut some of the development hurdles and really reduce the amount of waste potentially in the exploratory development phase. So I'm going to start by sort of reminding, I guess, that everyone knows biologics cost an absolute fortune, especially the APIs. You know, that anecdotally 1 gram can cost as much as 1.5 kilograms of 24-karat gold. So really, we don't want to be wasting any of this material. We want to try and make the most of it as efficiently and as effectively as possible. So I'm a real advocate for moving away from trial and error approaches, expansive DOEs, you know, really thinking about how we can use potentially new approaches, in silico tools and scientific rigor, a lot of the scientific knowledge that people in this audience have contributed to over the decades, to really see if we can shortcut some of and streamline some of the development activities that take place. So I'm going to jump straight into understanding the modality. I'm not going to explain what spray drying is. Most of you know that already. I won't go through the process, and I certainly won't talk about these are the different type of modalities. You know, I guess we've covered that over the preceding years and during these sessions over the last few days. Jumping straight into this, what we're going to do is we're going to use a case study. Aptar's working on a Fab, fragment of an antibody. And you can see here it's represented on the screen, the light chain, the heavy chain represented here. And just for people who are interested, it's a high affinity antagonist Fab, so it's used for obviously blocking the IL-4A receptor. And it's a challenging compound to work with, so I'm going to hopefully talk to you and convince you that some of the in silico tools that we've been developing really streamline some of the development program. So I should have really called this from angstroms to aerosols and going through the process. I'm going to start at the atomic level. I'm going to work my way all the way up to the aerosol, talking about the challenges, how they can be profiled and how we can mitigate them. So as most of you know, there's a number of chemical challenges we need to overcome first and foremost with proteins: deamidation, isomerization, oxidation, and the Maillard reaction. I should say my slides are particularly busy, so focus on the pictures and on the narrative, and I've kind of added the extra detail so people can look at them afterwards and refer to them. And then when it comes to the process as well, the sort of things that we're obviously overcoming, you've got shear-induced degradation, dehydration stresses, and aggregation stresses in the liquid and in the solid state that we need to think about and put in amelioration technologies for. So, starting at the atomic level, and we think about those individual atomic components and some of the residues, we can identify some of the core challenges with biologics just by looking at the amino sequence, identifying those pairs of residues that are likely to induce degradation. And so you can see here, look, I've identified a few of the degradation risks with deamidation, with isomerization and acidic cleavage. We can look for those particular sequences. But it's not just about the fact that they're there, it's where they are in the molecule, not just in the sequence, but actually do they have an impact on the secondary, tertiary, and quaternary structure? And really importantly, do they have an impact on the complementary determining regions? So if we look at the structure and the profile, it's just here as a map. I don't expect you to see all of the letters. And you can see I've highlighted those particular risk factors. And what I've also done is you can see, we can see the complementary determining regions outlined. So it's really, these are issues, and they're in the business end of the particular molecule that binds to the target epitope. So already we can see there's a particular challenge with some of these. But it's one thing to look at a 2D structure, it's another to start to profile this in 3 dimensions. So again, as we start to look at our in silico methodologies, we can start to think about, where these particular degradation routes occur. And again, once we start to apply the target epitope to our model, we can see really where the binding takes place and where the challenge is. And again, it allows us to map those particular residues and see, hey, look, they're pretty close to the business end of that particular molecule. So when it comes to the isomerization, what can we do about it? Well, we can start to think about already amelioration technologies quite quickly. No to phosphate buffers and acetate buffers, because of the huge swings potentially in the pH during the drying process. And obviously, once it's reconstituted in the airway surface layer, those microenvironments are going to cause some quite significant degradation issues. And yes, to histidine buffers and citrate buffers. And so if our modality already comes in one of these phosphate-based buffers, we're already starting to think about, look, let's get onto a dialysis, let's get onto a buffer exchange nice and quickly. Okay, on the oxidation risk, again, we can identify that that pesky sulfur atom here is going to cause us a lot of issues, but it depends. If it's buried deep within the structure, then it's not necessarily going to be exposed to those reactive oxygen species. If it's on the surface, then it is going to cause an issue. So using in silico modeling, again, we can identify the location of those methionine residues, and then we can determine that risk. So we've put them up here. We can see they're close to the CDRs, but actually are they close to the surface? Are they exposed? And so using something called solvent accessible surface area calculations allow us to determine whether or not those methionine residues are something to worry about. And the high SASA value sort of says, yes, this is a bit of a problem. And we can go further. We can do the calculations to look at each methionine residue, and we've got a problem in the heavy and the light chain. So we need to deal with the methionine residue straight away. So what can we do about that? So again, all in silico, we haven't gone into the lab yet. We know that we need to think about antioxidants as a strategy for this particular modality. But, as a starter for 10, adding more L-methionine into the formulation in order to act as a sacrificial lamb, ensure that those reactive oxygen species interact with those residues as opposed to the actual biological modality is a brilliant way to do it. And again, L-methionine's endogenous to the lung, so again, nice, safe strategy. And the other thing is we then start to think about the potential device and the packaging. We need to make sure that we're using aluminium blisters with low oxygen permeability, or the pucks that we use in the orbital device, all of which kind of prevent the moisture ingress and the oxygen ingress during stability. So we're thinking about the sort of device that's required for this particular modality too. Maillard reaction is pretty easy. Here, obviously, we've highlighted the lysine residues. They're everywhere, as you'd expect, and they're extending into the solvent interface. So, again, no reducing sugars, no lactose, glucose, or fructose. Yes, to trehalose, sucrose, and mannitol. And again, most of us know that, but it really kind of backs up the science behind it. But it's not just about what's chemically related. We need to think about the structure of the matrix as well, and we'll talk about that shortly. Again, looking at the modality itself and starting to do some in silico profiling, we're starting to think about domains of residues and the physical chemical properties they potentially impart on the areas around them. And obviously, one of the key ones is really profiling the hydropathy of various sort of areas within the structure. So here we can take our crystal structure again, our three-dimensional structure, and now we can apply a surface topology to it, and we can start to do some of the chemistry associated with the part that's obviously interacting with the solvent and interacting with the molecules within the neighborhood. And so we can use that surface topology to then think about where the hygroscopic and hydrophobic domains are within the surface and what potential challenges they cause us. So you can see here we've got cyan regions which are hydrophilic, and the maroon regions which are hydrophobic. And the issue really here is that the hydrophobic domains are going to move to the air, water, liquid interface of a droplet during the drying process. So we can see that, quite a lot of those are in the complementary determining regions. Those obviously are part of what's required for the selectivity and the docking of the target epitope. But we need to obviously deal with that. So adding something like leucine to our formulation is a real good first step that hopefully should make it to the surface before the modality. But let's prove that and let's work out how much leucine we might actually need. Here's whether or not this is an issue. So we've got something here which is a calculation known as a root mean squared deviation. And what it tells us, basically is just whether or not the crystal structure changes over time. And so how much it deviates from that original best practice, the original sort of structure that we've got here. And as our simulation goes through, for the normal molecule, it hits the air-water interface about 40 nanoseconds. And then essentially we start to see surface-induced denaturation taking place, where the surface tension rips the molecule apart on the surface, as we'd expect. When we add a certain amount of leucine, what we see is that the modality remains intact in its configuration, diverging very little from the original one very easily. So the leucine strategy is a good one to try. I guess then the question is, how much leucine? So we then need to think about modeling this in terms of thinking about surface enrichment as part of the solvent evaporation process and the film formation. And we can build a model that says, okay, let's test this. And we can do this in silico really, really quite easily. So here's our Z density profile, which is another silico quick tool. Here what it says is when we model this and we apply some leucine into our system, in 5 nanoseconds, we've already got this beautiful leucine shell appearing at the air-water interface, and the rest of the protein now is pushed back into the droplet. So for this particular modality, this strategy is going to work brilliantly. Then the question is how much leucine? And so what we can do is we can use a quality by design approach. We can calculate how much we need for a 3 micrometer diameter particle. We can work out the droplet that's required from an inflation perspective, and we can work out how much we need for surface coverage and create a bit of a safety margin, and then we can test whether or not that works. So here we've tested 5, 10, and 15% leucine. Too much, and we'll see a lot of kind of cohesivity on the surface of the particles. Too little, we won't occlude the surface, and the biological modality will fall apart. So at 10%, here's our surface tension kinetics validating model. Here you can see that the surface tension drops really readily in the first sort of 5, 10, 20 nanoseconds, plateaus out at 30, and at 40 nanoseconds, we've got complete coverage of that surface, and there's no change in the surface tension. So we can see that leucine is a great strategy. 10% works really well. It's the right sort of level. Again, we've all done this in silico in just a few hours, something that you wouldn't be able to do in the lab. And the added benefit of having sort of leucine is reduces the coefficient of friction between particles once they're formed, and therefore improves the aerosolization. So we're already into a really nice way of creating some good positive aerosol particles. Another in silico analysis here is the B factor, which is really kind of about how much the molecule stands up to the stresses. And we know that spray drying in particular is a particularly stressful process. High atomization gases mean that you're really putting that molecule under a lot of stress. So here we can see whether or not we should even do spray drying as a process. But thankfully, our modeling says that the majority of our modality is pretty strong and will hold up to the rigors, and we can test that at different atomization gases in order to make sure that's the case. At certain levels, we can start to see red appearing. This is showing the degree of flexibility. And obviously, as well as we start to see that white, we know that the complementary determining regions, the part of the molecule that's really focused on how obviously it interacts with that target, is also starting to be effective. So this also starts to then tell us something about how much aerosolization energy we need to put in, just enough to make a droplet of the right size for inhalation, not too much that we start to degrade the molecule. So already we've now started to come up with process parameters where the formulation parameters too. When we look at the electrostatic potential, this is really important. So we can map on dipole moments on the surface really quickly. And so we can see there's quite a few dipole moments, some areas of red and blue. And what this means is that in solution, we're going to start to see aggregation potentially occurring. There'll maybe be an increase in viscosity at higher increased concentrations, but ultimately we also need to make sure that we maintain that primary particle stability. Aggregation doesn't take place, and ultimately we can maintain the effectiveness of the modality. So again, we talked about histidine buffers being really important. And so here we can model how much of the histidine buffer is required in order to create that Debye shielding, neutralize the dipole attraction, and maintain the modality's individual primary stability. So we've done all this in silico. We've not actually gone into the lab yet. We don't need to do buffer exchanges and various other things. We're starting to think about a core formulation and a core process, all from the comfort of our laptop. Excuse me, I've got a terrible cold. So onto the stabilization. So when we think about the stabilization matrix, here what we're looking for is something that will confer a degree of stability to the biologic. And what we're going to do is we're going to strip off all the water, or as much of the water as possible. We want to make sure, obviously, that matrix confers that stability and ensures that the modality remains in its secondary, tertiary, quaternary structure on stability. If we use a crystalline matrix, so here we've agreed non-reducing sugar, that's simple. But if we think about a crystalline matrix as the drying front processes, what you'll see twisting, crushing, and deformation of the ternary and quaternary structure. And so we not only need a non-reducing sugar, but we need a non-reducing sugar that creates an amorphous matrix that flexes around the modality. And so we've got two hypotheses to work from, and we can model these. So we've got the mechanical entrapment, so essentially we encase the modality in a nice sort of amorphous matrix, but we have to make sure that the excipient of choice confers stability through strong hydrogen bonding points. In the second, with water entrapment, we leave a little bit of water on the surface that conveys that stability, and then we encase that water biomodality in that matrix itself. So in reality, it's a bit of both of those, but it's whatever it creates in order to create the largest Gibbs-free energy change between the unfolded, the active state, and the folded state. So back to our in silico modeling. So now we can start to think about our surface chemistry again. How much trailose do we need if that's what we're going to use? It fits the bill with being non-reducing sugar, lots of hydrogen bonding points, and forms an amorphous matrix, but how much do we need? If there's too much in there, then we're going to find it's going to start to crystallize potentially preferentially, affecting the stability of our modality. If there's too little, we're not going to ensure that the stabilizing effect is conveyed. and actually unfolding will take place. So using this model, we're able to identify those particular hydrogen bonding points in teal, and then we can do a quantitative calculation to get the precise excipient load. So what we found for this is that the hydrogen bond formation as a function of the molar ratio can be mapped. We actually found there's 558 potential hydrogen bonding points to 1. So 558 moles of trehalose to 1 mole of biological modality. And once you work that out, that's a 1.3 to 1 weight ratio. So 39% trehalose to 30% drug loading. Again, all done in silico before you've gone into the lab, you've identified the optimum point. That's really important because, again, amorphous matrices are unstable. You have to make sure there's just enough in there to have the stabilizing effect. If you don't, and crystallization takes place, or vitrification takes place close to the surface, then ultimately you will see solid bridge infusion, and you will lose the primary particle diameter that's required for deep lung deposition. So the stability of our amorphous matrices are obviously related to our glass transition temperature. The higher the TG, the better. We all kind of know that. But trehalose is inhibited a little bit, that small amounts of water with a glass transition temperature of about minus 135 degrees has a massive impact and drags that glass transition temperature down, therefore reducing the potential stabilizing effect of that matrix over the long term. So, we want as little water in there as possible, but we also have to acknowledge that some water will remain. We really need a better safety margin. So again, we use our in silico tools. We're here, we can profile a number of different excipients to add. It's not just about what will convey the greatest stabilizing improvement in terms of the glass transition. It's also about what will increase the viscosity of our feedstock. It's what will be miscible with the different excipients in there. And again, we can model all these, on our laptop nice and easily before we go into the lab and sort of waste any time doing expansive DOEs, looking at competitive experiments. And here we found one, ingredient X. I won't talk about any more than that at this stage. And it conveys a significant stabilizing improvement, pushes the TG up to 128.2 degrees. So a real boost in terms of the difference between raw trehalose and trehalose with this particular additive. Then finally onto the devices. So, we've created this fantastic formulation that's brilliant at aerosolizing and maintaining some stability. But in terms of creating a stable aerosol, that isn't easy either. These particles are small, and the small surface area to volume ratio has an impact on just how they flow, how cohesive they are, and the interparticular energies between them. And then once you get it moving and it's in flight, the physics of flight take over, and obviously those are pretty complicated too. So the point here is you need to find the right device for these formulations. You need to pick the right device from the off and not have a massive device screening program. Let's think about that. So for our devices, the things that we need a strong aerosolization energy, but one which necessarily doesn't require huge amounts of patient inspiratory breath in order to drive those. The patients we're talking about here have got compromised lung function. So we need to find a way to amplify or use, unique deaggregation methodologies in order to really drive the energy into the right place of the formulation without necessarily causing a significant draw on the patients. And then we need to be able to deliver large volumes, quite a lot of these biological modalities, in particular this particular MAB. We need to obviously deliver significant amounts of powder. So again, high payloads are really important. And crucially, as I said at the start, making sure you've got the right container closure system, blisters or pucks, potentially protected with Aptar's CSP technology, are all advantageous for these formulations. So very quickly in summarizing, to put it all together, we've got our biological modality, we've got a little bit of water remaining, we've thought about the microenvironment, the pH, in order to protect it in its liquid state, but also in the solid state as well. And we've thought about having antioxidants or L-methionine in order to protect against reactive oxygen species instability. And then we've got our primary stabilization layer. Our excipient is a non-reducing sugar. That one's obvious. It's got high hydrogen bonding potential. It creates an amorphous matrix and works with the water in order to stabilize that. And we've got a couple of stabilization layers. And then when we've got a small amount of additional stabilizing excipient, we've boosted that with a TG enhancing additive already from the start. We've selected that additive because it fits well within the formulation. And then on the particle surface itself, it's coated with leucine. The leucine helps to maintain the biologic within the droplet, keep it encased and keep it protected. It prevents, obviously, from expressing on the surface, and then has the added benefit of improving the aerosolization prospects once we obviously put that into a device. So that's the most basic form. But what's really important, I think, is that almost all of these things we've done in silico. We haven't gone into the lab yet. We haven't done DOEs. We haven't gone through a trial and error process. So it's not to say that this is a perfect formulation, but it's a brilliant place to start. And we've done it by reducing the number of experiments. So just like, or obviously conversely to Goldilocks, we don't have to break a lot of chairs and waste a lot of porridge. We can find the perfect zone in silico and do it relatively quickly using some of these tools and scientific data and strategies. And I think for us, it's more streamlined, more cost effective, more time effective, and ultimately better and more akin to the spirit of quality by design moving forward. So quite a lot of papers and references here, obviously, in order to put this together. And thank you very much for your attention. Really appreciate it. And I'll take any questions. And my apologies for talking so quickly. There was a lot to cover, but thank you very much for your attention. I appreciate it.
Well, thanks very much, Dave. That was a fascinating talk and a huge amount of information to try and cram into about 20 minutes.
Sorry, Dave.
Does anyone in the audience have any questions? Any questions for Dave?
I guess.
I think there's a hand at the back there to the right.
Hi, Leonidas. Also, very nice presentation. Thank you. I have a question regarding the stability of your formulation. Since you introduced like 10% leucine in your formulation and it solidifies in a mostly crystalline state, do you see long-term stability issues if you have nucleation already throughout the leucine, which subsequently will also crystallize the rest of your particle?
Yeah, it's a really good question. This is why the leucine concentration is really important. If you've got too little, you don't really get the surface occlusion you expect. If you have too much, then you don't necessarily get it all expressed on the surface. And what you should see is a two-phase system where the leucine is distinctly separate from the trailose, and provided it's reasonably separate and the concentration gradient is less beyond the interface, then yeah, you shouldn't see seeding of crystallization, especially over the long term. And most of the formulations, we don't really see that. Things like XPS and SCM have shown real good differentiation between those layers in previous projects. And so actually you don't see the seeding of crystallization you may expect. It really is about making sure that you calculate the right amount. If you don't too much, then you will obviously see that impact.
Thank you.
Oh, thanks.
Got any other questions?
It's so hot. Yeah, at the front, just at the front.
Yeah, thank you, Jose Maria Langaron from BioNanoPharma. Very, very good talk. Thank you. So this is model for a spray drying process. So how is the particle size affecting this? And can you also model other type of dryings like electrostatic forces?
Yeah, sure. I mean, look, these models are really simplistic to do. I mean, some of the data there really takes around a couple of days to do for a modality. And then you have to think about, obviously, the physical process. Spray drying is the one I know the best, so I've used that. But you're right, you can put in these models to look at any particular drying process, provided the physics is particularly well understood. There's a degree of assumptions. It's not a super hyper-accurate model. It's designed to identify risks and to ameliorate them. But yeah, other drying processes can be used, and it's a case of just applying them. Yeah, it's pretty versatile.
I've got a super quick question. Okay, sorry, over the floor, thank you.
Dave, this is Geraldine Ventoy of Philips Medisize. It's a great talk, and it really does exemplify how complex, biologic formulation can be. I think what surprises me is you've got quite a ways down in terms of modelling and hypothesis and introduction of lots of different critical criteria in the formulation before you actually make anything. As we all know, hypothesis, when it works out, it's great, but lots of things can go on. And do you not worry that, once you start making these things and you start seeing unexpected results, you wouldn't be able to deconvolute out what the leucine was doing, what the various, strategies that you're using to actually get to the ideal situation? And should you actually stop, check your hypothesis is working, so you can really see each parameter and do, statistics and some form of factorial analysis to understand really what's going on?
Yeah, sure. And to be honest, these models are, use them for approximations to try and streamline and identify core areas and core risks. But yeah, as you go along, you do experiments, you do studies. We've got experiments running with this particular system now, and it's working really, really well. You have to do that cross-validation. I've made it a coherent story. It's not exactly like this, but actually the more and more experience you get, and you show that the models have real good parity to experimental truth, and again, modalities are slightly different, then it gives you confidence to do more and more of the modelling up front from a quality by design perspective as opposed to down the line. I'm a massive DOE proponent, but I think it's really important that where you start and you create that center point. It's about not having expansive areas and panning for gold. It's about zooming in on a smaller region so that your DOE is less about 15, 20 runs, and more about, something smaller, 9 runs, and optimization, for example, where the small amounts of variability that the model can't necessarily compensate for. You can put more and more and more effort into your modeling in order to create, increased precision. But fundamentally, it's about identifying risks and being able to ameliorate those risks, things like the chemical degradation, things like how much leucine and how much tralose. But again, you can tweak that once you get into the experimental area. It's really about preventing the runs where you don't get much from, things where they don't work, reducing that and focusing on the real sort of area and the real operation space that you do really want to profile as you move through the development process. So I hope that answered your question, which is keep it vague and high level and quick. Don't necessarily go into the detail and then assume that it's going to tell you all the answers. That's not really how this works.
Great. Okay, well, thank you, everybody, and thank you particularly today for a really good talk, and I'll hand over to Michelle to introduce the next speaker. Thank you.